In the previous lecture, we went over the carbohydrates and the nucleic acids, and we practiced identifying them. And in this lecture, we're going to go over the lipids and the proteins, and we'll practice identifying those as well. So there are certain things about lipids you need to know. One is that they're the only of the four groups of macromolecules, the lipids is the group that doesn't have any polymers. So we really don't talk about the monomers and the polymers. We will talk about components of lipids. The main thing about lipids, the thing that makes them all in one group, is that they're all hydrophobic molecules. And you'll recall that hydrophobic molecules are the ones that have mostly nonpolar bonds, and usually that means they're primarily made of carbon and hydrogen. So lots of hydrocarbons in the lipid group. And you won't see very many functional groups in the structures. So biologically, the ones we will talk about are the fats, the phospholipids, and just a tiny bit about steroids. Mainly we're going to focus on fats and phospholipids. So fats are molecules that are made up of smaller components, and the smaller components of a fat are called glycerol, which is a 3-carbon molecule that has 3 hydroxyl groups, and some other molecules called fatty acids. Fatty acids have a long hydrocarbon chain and a carboxyl group attached to them. So you're going to have to be able to identify glycerol from an image, and we'll practice that in a minute. You're going to have to be able to identify a fatty acid, and then when you put it all together, the whole molecule, when all those parts are connected covalently, is called a fat. So the fatty acids, typically you have 3 fatty acids that are connected to one glycerol molecule. And when that's all connected together, the molecule can usually be called, it can be called fat, it can be called a triacylglycerol, or it can be called a triglyceride. So those 3 words are synonyms. And the fats are hydrophobic. Even though you do have that carboxyl group in the fatty acids, there's so much hydrocarbon in a fat that that's the predominant part of the fat. It makes the whole molecule hydrophobic. Okay, so here's where we look at the components. So you want to get used to looking at how they look in the molecular structures. So the glycerol is a 3-carbon molecule, and it has 3 hydroxyl groups. So you can see 3 carbons, and then here's the 3 hydroxyl groups. And then everything else is just hydrogen. All right. The fatty acid, there's different variations of fatty acids, but they all have a carboxyl group. Now you'll notice this carboxyl group is sort of been rotated, and it's sort of pointing, I guess, towards the left. And then, so that's the carboxyl group. And then the rest of this is just hydrocarbon. So there's a lot of this. Now the fatty acid, that can be longer or shorter than this, but they all are called fatty acids. We're not going to get into the super specific names of the fatty acids. Each one has a different name, of course. And so when the hydroxyl group of a fatty acid interacts with the hydrogen part of one of these hydroxyls on the glycerol, so they've kind of highlighted for you in pink, this OH in the fatty acid and this H in the glycerol come together to form water, and the water, which is not shown here but it should be, is released, so that's called dehydration synthesis. And the dehydration synthesis connects the fatty acid. In this case, it connected this fatty acid to this part of the glycerol. And then if you have 3 of these fatty acids, then all 3 of them get connected. One of them gets connected to this part of the glycerol, and one of them gets connected here. And so if you have 3 fatty acids, all 3 of them get hooked up to the glycerol. That's where you get the triacylglycerol. And then you would release 3 waters as a result. The bond that holds each fatty acid to the glycerol is called the ester bond. It's this bond here. It's called the ester bond. I wish my mouse didn't do that. I actually think it's the software because I've traded out different mice and it still does it. All right, so that is the ester bond is what holds the fatty acid to the glycerol. So when those 3 fatty acids are bonded to the glycerol, then we call it a fat, or we call it a triacylglycerol, or we can call it a triglyceride. So the main variation in fats has to do with what kind of fatty acids they contain. And there's really 2 kinds of fatty acids, saturated and unsaturated. And when we talk about saturation of fatty acids, what we're talking about is how much hydrogen the fatty acid is holding. So you remember the hydrocarbon portion of the fatty acid. So we look at that. We don't look at the, all fatty acids have this carboxyl group, so don't look at that. But in the fatty acid, in the hydrocarbon portion of the fatty acid, I put a box around this one, and then I'll put a box around this one because this one's a little different. All right, if the hydrocarbon portion of the fatty acid doesn't have any double bonds, no double bonds, just in this part, then that's saturated. It means it has as much hydrogen as possibly could be. If it does have a double bond, it could be more than one, but you can see one here in the hydrocarbon portion, then it's not holding as much it's not holding as much hydrogen as it could. You can kind of see there's sort of like empty spots here and here where there's no hydrogen. And it's because of the double bond that carbon can't hold as much hydrogen. So because of that, it's not better or worse. We're just saying that it's different. So when there's a double bond in the hydrocarbon portion of the fatty acid, we call that unsaturated. It's not completely saturated with hydrogen. That's what we mean by saturated and unsaturated. Saturated with hydrogen, holding as much hydrogen as it can or not holding as much hydrogen as it can. And that would always be due to a double bond in the carbon backbone. Okay? So here's a close-up of that saturated fatty acid here. All right, this one they turned it around, so you've got the carboxyl here, and then where you're looking, if I gave you this picture and I said, is this saturated or unsaturated, you're looking at the hydrocarbon portion and looking for any double bonds in that part. Don't look at this double bond because all fatty acids have that double bond. But look in the hydrocarbon portion. So this one has no double bonds, so it means it's saturated. It has as much hydrogen as possible. And one thing about saturated fatty acids is they will form these straight chains. So like the shape of them is somewhat like the way the picture looks. They form these straight shapes. And that means they can all stack up and pack very tightly in a solid. And so they form a solid at room temperature. So something like butter or fat or lard, meat, fat from meat, is all solid at room temperature. Like if you have a room temperature piece of steak and it has fat on it, the fat would be, white. It would be that solid form even at room temperature. Now, yes, you can cook it and it'll melt, but we're talking about room temperature. At room temperature, saturated fatty acids will be solid. And these are the ones that you don't want to eat in your diet because they tend to pack together. They can block your blood vessels. So they're not, they cause some health issues. So meat fat is, butter, fatty meats, lard. That's why that they cause trouble, is that they have these straight structures and they can pack together and form these solids. Now, unsaturated fatty acids. So here's an example of an unsaturated fatty acid. You don't need to memorize the specific names. But this one is unsaturated, and the way you would know it is you're going to look just at the hydrocarbon portion. Again, don't look here because all fatty acids have that double bond. But in the hydrocarbon portion, you do have a double bond, and so that means it's unsaturated. If it has one double bond, we call it monounsaturated. You remember your prefixes. And then usually they don't, I don't know why they don't use di-unsaturated or tri-unsaturated. Usually it's either mono is one, and they use poly for anything more than one. So it's kind of weird, but that's the way the language has come to be used. So I didn't make that up. I didn't make the decision, so we just have to go with the way that it already is. Now, at room temperature, unsaturated fats, because in this picture they look like they're straight, but in that other slide it was more realistic where you would have a bend. The double bond creates a bend in the molecule, and that means that the molecules have trouble packing tightly together because they're all kind of bent up. And so they can't pack that tightly, and you probably know that in a liquid the molecules are more loosely arranged. In a solid they have to be very tightly packed. So they're not able to pack very tightly, so they can, at room temperature, they will stay in a liquid form. So fats and oils are really no different, other than the fact that something that we call an oil is just an unsaturated fat. So we commonly refer to them as oils, but fats and oils are both triacylglycerols or triglycerides in their structures. The difference is, do they have double bonds in the hydrocarbon portion of the molecule or do they not? And the unsaturated fats are considered healthier because they don't pack together as much and cause blockages in your blood vessels. Okay, so you're going to need to be able to identify. Let me take a break from the lecture just for a moment, and I'm going to have you look at your packet, and let me bring that up. Okay, so let's look at this. This is the third page in your Chapter 3 packet that I posted on eCampus, and here's some examples. So can you pick out on this, so I have them labeled A, B, C, D, E, F, G, H, I, J. So can you find first where the fatty acid is? One of these is a fatty acid, just a fatty acid. So look at it, maybe pause the lecture and see if you can find the fatty acid before I give you the answer. The answer is G. G is the fatty acid. So they've drawn it in actually a fairly realistic drawing here where you can see many bends, but this is a hydrocarbon chain. And then on the far right side you see the carboxyl group. So G is a fatty acid. And then I might ask you, is this saturated or unsaturated? In order to figure that out, you're going to look at just the hydrocarbon portion and look for any double bonds in it. You have 3 double bonds. So this would be polyunsaturated if we were getting picky, but it's definitely unsaturated. So G is an unsaturated fatty acid. The other thing I could ask you to look for is a fat. Now a fat has that glycerol, remember that 3-carbon structure with 3 hydroxyl groups, connected to 3 fatty acids. So look carefully and see if you can find the fat, the triacylglycerol? The answer is J. J is a triacylglycerol, or fat, or triglyceride. You can see on the left side the 3 carbons of the glycerol component, and then the 3 chains hanging off of it. Those are the fatty acid components. And they're all covalently connected together now by ester linkages. And so the whole thing, now it's one molecule that we call fat. You might have been looking at C, and C is something we're going to talk about. But the reason C is not strictly a fat is that you'll notice that on this first carbon, there's a phosphate group and there's some other stuff here, so it looks kind of not quite right. Now on the second carbon and the third carbon, you do have fatty acids. You have one that's saturated and one that's unsaturated. On J, you have 3 that are saturated. But anyway, C is an interesting one, but it's not purely a fat. So J would be the fat. All right? So G and J. G is a fatty acid, J is a fat. And fatty acids are part of the components of a fat. Let me go to the next one. This slide, this one is pretty good too. I call it more practice. All right, now if you can do this one, let's see. Let me ask you to look for a fatty acid on this picture. Now you're looking for a carboxyl group attached to a long hydrocarbon chain. So there are 2 on this image. Is there anything down here? No. There's 2 on this image. Number 9 is a fatty acid, and number 17 is a fatty acid. You might be lured by some other things like number 1, except the problem with number 1 is this is not a carboxyl group. This is a C double bond O, but this is not an OH over here. It's just an H. So that's just a carbonyl. It's not a carboxyl. The other thing that's wrong with it is this chain behind it, this carbon chain has all these hydroxyls. You're not allowed to have those in a hydrocarbon chain. Hydrocarbon chain has to be pure carbon and hydrogen. And for the same reason, number 4 looks like it could be, looks similar to a fatty acid, but you have, at the end here, you have just a carbonyl group, and then below it you have a chain of carbons, but it's not a pure hydrocarbon because you have too many hydroxyls. In fact, number 1 and number 4 are simple sugars in the linear form. I won't give those to you though to recognize. I would give you like number 6 to recognize a sugar. So 6 is glucose. Do you still recognize it? All right, number 12 is a simple sugar called ribose, a 5-carbon sugar. Number 15 is a disaccharide. So try to look for some of those things you already know, the sugars. All right. But the fatty acids are number 9 and number 17. Now, can you find a fat, a triacylglycerol? And that would be number 16. Number 16 is a triacylglycerol. It has the 3-carbon glycerol, and then it has attached to it 3 fairly short hydrocarbon chains. So what is number 10? Look at number 10 and see if you can tell me what that is. It's a 3-carbon molecule with 3 hydroxyls. It is a component of fat, but it's not the fatty acid part. It's the glycerol. So number 10 is glycerol. Now the nice thing about this is, as I'm going through this, you can mark down what everything is. Then that'll be your answer key. And then you can print out a blank copy and see if you can figure it out from start to be able to recognize things on your own without looking at the answers. That's the goal. That's what we want to get to. And then for each of these molecules, you need to know the characteristics that I'm going through in the PowerPoint. Okay, so let's talk about phospholipids. The name of it kind of gives it away. It is a lipid. It is going to have fatty acids, but it's only going to have 2 fatty acids. It's basically like a fat, because remember, a fat has a glycerol backbone and 3 fatty acids. A phospholipid is like if you took a fat and you took off one of the fatty acids, and instead you attached in the place of the fatty acid, you attached a phosphate group. Then you would have a phospholipid. There's different kinds of phospholipids, but they all have the general structure shown here. The R is the thing that could be different on some different phospholipids. This letter R at the top of the structure molecule here. So sometimes you'll have like a little nitrogen or a little amino group or something like that. You always have, though, the phosphate. That's the giveaway. And you have basically, and this one is drawn sort of flip sideways, but you got to get used to whichever direction they draw it. They can rotate it anyway. But this is your glycerol backbone. These are your 2 fatty acids. And look what they did. They got really lazy. Instead of drawing the hydrocarbon, they just drew a zigzaggy line. Remember I said anytime there's an angle or a corner, that's assumed to be a carbon. So each of these points on the zigzaggy line is a carbon. And then they don't even draw in the hydrogens. They assume you know that carbon forms 4 bonds, and so whatever number of hydrogens you would fill in to make that happen. On this one, they give you an unsaturated, so they show like a little double line on the zigzaggy line, and that would indicate a double bond. And they show it bending. It bends the molecule, so that's a reasonable way of drawing an unsaturated attached fatty acid. But the whole molecule, this is really now the fatty acid components of the larger phospholipid molecule. But the phosphate group is a good giveaway for you, and that's in the name, phospholipid. All right, so it's basically a fat. It's basically a triacylglycerol. I don't know what I'm doing there. And instead of having 3 fatty acid chains, it only has 2, and then in the place of the 3rd fatty acid chain, there's a phosphate. And you'll see how the phosphate kind of flips up. The fatty acid chains face down, and the phosphate part is sort of flipped up. And that creates something really interesting, because you remember that phosphate is acidic and will ionize. It means it takes a charge, and that makes it very hydrophilic. And so this part of the molecule, the part that has the phosphate, is very hydrophilic. But the rest of the molecule is very hydrophobic because it's all hydrocarbon. And so you end up with it's a very large molecule. I mean, these are macromolecules now. So when you have macromolecules, you can actually have part of the molecule, like this side of the molecule has one behavior chemically, in this case hydrophilic, and this end of the molecule has a different behavior, hydrophobic. So this is really special. You have a molecule that is all one molecule, but one end of it is very hydrophilic and one end of it is hydrophobic. We usually call the end that's hydrophilic the hydrophilic head, and we usually draw it as a circle like they did. And then the hydrophobic parts, we call them the hydrophobic tails, and we draw them like 2 little sticks. So this drawing, because this molecule is so large, we start to just do a drawing like this, and anytime you see like a circle with 2 sticks coming out of it, that means a phospholipid molecule. All right. So, but you will have to be able to recognize the detailed structure as well. All right. And when a molecule is so large that it has some hydrophobic regions and some hydrophilic regions, we call that kind of molecule amphipathic. Amphi meaning both. It has both hydrophilic parts and hydrophobic parts. So let me show you the practice packet again and see if we can identify the phospholipids. Let me go up to the 3rd page again. Remember I said C was kind of attractive when we were looking for just a fat? But the thing about C is it has, oh, I think I have a pen here. Let me see if I can use my pen. No, what is that? Yeah, I don't want to do that. On C, what you have is, you can see on the, and I copied this from somewhere that had green background. So on C, you can see the carbon, carbon, carbon of the glycerol component. And then down here, you have 2 hydrocarbon chains. So those are the tails, so to speak. And on this drawing, they didn't flip the phosphate over, but it doesn't matter. This is the phosphate group, and this is the R group. Remember I said there would be maybe something else there. So this phosphate group, though, is enough to say, okay, this is a phospholipid. So C on this page is a phospholipid. And then when you come down here on the 4th page, look at number 5. I mean, first of all, it's huge. But this one is drawn more like having the head at the top here. You can kind of draw a circle around this part at the top, and then 2 tails hanging down. It's more of a giveaway in that sense. But you can see the phosphate here, the 3 carbon backbone of the glycerol, and then the 2 long fatty acid chains. One of them has a double bond, so it's bent. So that's a phospholipid, number 5. You can see how large these molecules are getting, and so that's why you can have one region that's hydrophilic and a different region that's hydrophobic, because they're so large in the sense of molecules. All right. All right, so let's go back. Oops, not there. To our lecture. All right, so here's 3 ways that phospholipids could be drawn. You can see the actual molecular structure that's drawn here. And that's kind of what I'm asking you to see in the practice packet for identification of the structures. In the middle, this is called the space-filling model. You probably won't see that very often, but usually we draw the phospholipids with the symbol on the far right. We have a little circle for the head, 2 little lines for the tails. So you do need to recognize the structure, but it could be drawn just simplistically like in part C of this image, or it could be drawn out with the more detailed chemical structures like A. So you kind of be able to recognize it. So when you have a lot of phospholipids together, we use that little sketch. We use the circle with the 2 tails. So the hydrophilic heads and the hydrophobic tails. I don't know why this is. Let me fix this. There's a little issue here with the... Let me send that to the back. There we go. So when you put a whole bunch of phospholipids into water and you shake it, they will automatically self-organize into one of 2 structures. One's called the micelle, but the one that's important for biology, for cellular structures, is called the phospholipid bilayer. So that's the one we're looking at here, the phospholipid bilayer. And they've just shown like a little chunk of it. But the phospholipid bilayer it actually forms the main component of a cell membrane. We call it the plasma membrane. And so you take this concept here, this little chunk of picture, but you extend it and you curve it, and you curve it all the way around the cell in 3 dimensions. You would get the main component of the cell membrane. The hydrophobic tails point towards each other. So you have one layer of phospholipids with the heads pointing up, so to speak. And you have one layer of phospholipids with the heads pointing down, so to speak. And so the tails are playing footsies in the middle. And that creates a pretty thick layer that's all hydrophobic. And so what that creates is a boundary between the outside of a cell, which is mostly aqueous, mostly a water-based environment, and the inside of the cell, which is also mostly a water-based environment. But this creates a hydrophobic layer, a hydrophobic boundary between what's on the inside of the cell and what's on the outside of the cell. So the water on the inside of the cell cannot cross that hydrophobic boundary. And the water that's outside the cell can't cross it. So it keeps what's inside the cell in, and it keeps what's outside the cell out. And it's a fundamental structure of cells, not only the plasma membrane, but in eukaryotic cells, you have smaller little membranes inside the cell that create little compartments as well. And it keeps things outside of that compartment or inside of that compartment because it has that same hydrophobic layer, that same boundary. So phospholipids are important for membranes of all types. Now, steroids, I'm not going to talk too much about steroids, but they are included in the lipid group, and they are not structured at all like a fat. So fats and phospholipids have a certain similarities in structure. Steroids are not structured at all like them, but they are mainly hydrocarbon, and for that reason, they're very hydrophobic. And interestingly, cholesterol, here's cholesterol at the top. Remember, these ring structures, every time you have a little corner or a little intersection of lines, that implies that there's a carbon there. And then anytime you have a carbon that doesn't seem like it's making 4 bonds, it is. They're just not showing the hydrogens that make those additional bonds. So not all hydrogens are drawn into the structures, but all these little points and corners are all carbons, and there's tons of hydrogens that are connected in there. And so you have this structure of cholesterol. And cholesterol is actually an important part of it. There's a lot of cholesterol that's inserted into that hydrophobic tail portion of the cell membranes. So cholesterol is a component of the membrane. The main component is the phospholipid bilayer, but then you have other things that are kind of stuck in. And one of those things that's stuck into the membranes, there's cholesterol all over the place. And cholesterol is also the main component of hormones. So your body takes cholesterol. Not only does it need it in the cell membranes, but it converts cholesterol into the hormones that your body makes. So most of your hormones are steroid hormones. That means they have a basic structure starting with cholesterol that gets modified and to make it into a hormone. What's modified is whatever's attached on. So the main structure of cholesterol gets changed into, you can see some differences in the attachments. And that now it has a little bit of a different shape, and that gives it a function in the body, the hormone. So hormones, most of your hormones, natural hormones, are steroids. You do have some hormones that are not steroids, like insulin is a non-steroid hormone. Anything that's non-steroidal, it means it doesn't have a shape like cholesterol. It's not based on cholesterol. But a lot of hormones are steroids. Okay, the last group, so we went over lipids. Now we're going to look at proteins. All right. Proteins are, in reality, the most diverse group of the biomolecules. There's a lot more kinds of proteins than any other class. So there's more kinds of proteins than carbohydrates, than nucleic acids, than lipids. They're very diverse. Basically, proteins make everything in your cells, or they are the structures in your cells. Proteins either are the parts or they make the parts that your cell needs. So some of the parts are structural, some of the parts are enzymatic, but everything depends. All the other groups are important, but if I had to pick one that was the most important in the cell, I would pick proteins. Because proteins do everything. They do everything, they form everything. It's just a super critical group. Now, you don't have to memorize this list, so don't memorize this list, but there's tons and tons of proteins that we will learn some of them later on, just a couple, and they can become examples, as you add to your repertoire of knowing what certain proteins, what they actually do. But there's just tons and tons of proteins. So a key thing that proteins do is that proteins are act as enzymes, and enzymes speed up chemical reactions. So we will be talking a lot about enzymes. And a lot of enzymes are proteins. There are some enzymes that are made of RNA. It's actually not that unusual. And another interesting thing is there are some enzymes that are a combination of RNA and protein together. So you have some enzymes that are purely protein, some enzymes that are purely made of RNA, and some enzymes that are made of both combined. We're going to talk just kind of right now about the ones that are just purely protein. But they speed up chemical reactions. That's what an enzyme does. So the protein enzymes speed up reactions, right? But we're really going to focus on the structure in this chapter. So the monomer unit that makes up the polymer, the monomer of proteins are called amino acids. And amino acids, I think what students get confused is there's a different group of molecules called nucleic acids, but amino acids don't belong to that group. Amino acids belong to the protein category. So be careful. Nucleotides make up the nucleic acids. And I know it has the word acid, but amino acids make up the proteins. So make a point to keep that straight. Okay, because that's very fundamental. All right, very fundamental that you can just, if I say, okay, what's the monomer unit of all proteins, you can just rattle off amino acids. That's where you want to be. You want to be able to just rattle it off, okay? So you got to practice that. The fundamental structure of all the amino acids, there's actually 20 different amino acids, although all 20 of them have the same basic structure. We usually call it the backbone structure. All the amino acids have, and this is drawn in a certain way, but in the way this picture is drawn, on the left side you see the amino group, NH2, and then on the right side you see the carboxyl group, C double bond O OH. So you should recognize those. All amino acids have those two groups. They all have a carbon between those two groups. So it's really a small chain that goes N, C, C. That's kind of the backbone. And then this carbon in the center is called the alpha carbon. The alpha carbon always has one hydrogen attached to it, and then something else. R means something else, the rest. The side chain, sometimes we call it the variable chain. Sometimes we just call it the R group. So R group, side chain, variable chain, all means the same thing when we're talking about the amino acids. But the rest of the structure is consistent from one amino acid to the next. What varies is what the R group is. When you take biochemistry, you'll have to memorize the R groups. It's really boring, and it just takes a lot of brain space. But you don't need to memorize them now, okay? So there's 20 of them, and in this picture they've highlighted for you the R groups in blue. So you can see the different R groups, and depending on what the R group is, the amino acid gets a certain name. You don't need to memorize the names. Most of the names end with the letters I-N-E. There's a couple exceptions, but most of them end with the letters I-N-E. So you have glycine, alanine, valine, leucine, methionine, isoleucine. But the thing about the R groups is, because of the R groups, some of the R groups are nonpolar, some of them are polar, some of them are fully acidic, and some of them are fully basic. And so the R groups actually give each amino acid a little bit different chemistry, a little bit different chemical properties. So the R groups, each of them is kind of different. And what a protein is amino acids joined together by dehydration synthesis. And when the amino acids are joined together, they're always joined together in this way. The hydroxyl portion of the carboxyl group on one amino acid interacts with the hydrogen portion of the amino group on the next amino acid. And that's where the water comes. Again, they didn't write the water, but the water is released. And then that creates a bond directly between the carbon of the carboxyl group, which is now not a carboxyl group anymore, and the nitrogen that was an amino group, but now it's not an amino group anymore. Not really. So you have that. That bond is called the peptide bond. All right. So the glycosidic bond is the bond between the sugar monomers. The ester bond is the bond between the components of the lipid, the fats. The peptide bond is the bond between amino acids in the protein group. Oh, and I forgot to tell you, the name of the bond between the nucleotides in DNA and RNA is called phosphodiester. I better write that down. So the bond between nucleotides, this is a throwback to the previous lecture, but I don't think I mentioned it. Nucleotides, there's a bond between nucleotides, and that bond's called phosphodiester, which is really phosphodiester. So you have the ester bond in the lipid group, and you have the phosphodiester bond in the nucleic acid group. So if that's not going to confuse you, I don't know what is, so make some flashcards, right? But in the protein group, back to this slide, the bond between amino acids is the peptide bond. And what you can see is after you have, for example, these two amino acids joined together, then another amino acid can come in and interact in the same way. The amino end of another amino acid, here it is, that means amino acid there, then that can interact and they can keep joining like trains, hooking together, cars of a train, all latching together from, as long as the amino end of one amino acid interacts with the carboxyl end of the next amino acid, they can form that peptide bond. So proteins, like all of the polymers, they can be any length. And so sometimes when we have a long chain, we call it a polypeptide, or you can call it a protein. All right. So polypeptide specifically refers to a single chain. Protein is a little less specific. It can refer to a single chain, or it can refer to several chains that are wrapped together. So if you say polypeptide, that implies just one long chain of amino acids. If you say protein, it could be one chain or it could be a couple chains that are wound together. The shape of proteins is very important to their function, and proteins will fold into three-dimensional shapes. Frankly, all of the molecules have shapes. Carbohydrates have shapes. We talked about the spiraling shape of like starch and the straight fibrous shape of cellulose, for example. DNA has a shape. It forms a double helix if it's double-stranded. And the proteins have shapes. So all molecules have shapes. It's just that protein shape is something that we focus on quite a bit. And it has to do with, remember I said that each of the amino acids that's joined into this long polypeptide chain has a different R group. Well, there's 20 different ones. So you have this chain of amino acids, but you have all these different R groups, and some of the R groups are hydrophobic, and some are hydrophilic, and some are fully charged. And each protein, depending on the order in which those R groups exist on the chain, will fold into a certain shape. It's actually amazing because the shape is pre-programmed into the way the protein is built has to do with the order of amino acids that are linked together, and that dictates it to go into the correct shape. It's actually fantastic if you study protein kinetics, but folding kinetics. But anyway, don't worry about that. But the level of structure in proteins is usually broken down and called, we refer to the levels of protein structure as primary, secondary, tertiary, that means third level, and quaternary is fourth level. And what that means is when you have the amino acids in a long chain, that's called the primary structure, so the straight chain. But it doesn't stay straight chain. okay, because it actually folds. So some parts of the protein might actually spiral. There are some proteins where the whole protein spirals, or it could just be a section within the protein that makes a little short coil. Some parts of the protein will fold back and forth. That's called pleated sheet. And so typically on a long protein, you'll have like a region within the protein. Let me see if I can draw it, because I don't think this picture really gets to it. You would have like a region, like here's the protein, here's the straight chain, and then you would have a region that coils, and then it stops, and then maybe a region that folds back and forth, and then it stops, and maybe another coil. So within the larger protein, you have these little sections of secondary structure. The secondary structures are these coils and these folds. So that we usually call the coils are called alpha helix, and the folds are called pleated sheet. All right. And so the helix and the sheets, those are smaller sections within the larger typically. Now the third level of protein structure is if I take that whole chain and it's almost like I crumple it up. There's really two main types of tertiary structures. One is just a very crumpled up shape. It's irregular, and usually we call it globular. So the picture on this screen is would be an example of globular. So globular just means, like, I usually describe it as like the shape of popcorn when you pop it. It's, you know, popcorn has a three-dimensional shape. It's kind of blobby, so, and it has little niches and little things, but each kernel of popcorn has a unique three-dimensional shape, but there's not a geometric term that can describe it. So we call it irregular. But it's a three-dimensional, irregular, globular shape. Most proteins have that. The only other shape proteins will tend to take would be fibrous, so long and straight. So tertiary is the shape of the whole protein, whereas secondary structure is shapes within the longer strand. All right, so primary is just the order of amino acids on the chain. It's not really so much of a shape. It's just a straight chain concept. Secondary is the regions that will spiral or form the sheets. Tertiary is the whole shape of the whole chain. Quaternary is when you have more than one chain that sticks together. Remember I told you sometimes you have more than one polypeptide that wraps around each other. That's called quaternary. Not all proteins have to do that, but a lot of them do. So you need to memorize the definitions, just roughly speaking, the definitions of each of these, okay? So the primary protein structure are the amino acids in the chain without any particular shape. Secondary structure really is small regions within the primary structure that either fold or coil. Tertiary structure is one polypeptide fully folded. Quaternary structure is when you have more than one polypeptide attaching to each other. Okay? Now, when a protein loses its shape, when it loses its quaternary or really tertiary structure, when it unfolds, it also loses its function. So we call that a denatured protein, an unfolded protein. So in the picture you have a properly folded protein, the way it would exist in a live cell. But the way that you can unfold a protein is heat it up to boiling. Extremely high temperatures will unfold proteins. Or drop it in acid, or drop it in a strong base. So large changes in pH and heat are the main things that will unfold proteins. Some proteins can renature. That means, like, if you heat them up, when they cool down, they can go back into their shapes. Some don't. And most don't, but some do. For example, when you cook an egg, what you're doing with the egg white is mostly protein. It's albumin. When the egg white is, when you first crack open the egg, all the albumin in the egg white is folded. And it's clear in the egg white. And then when you cook it, it turns white. And that's because you've denatured all of it. And that denaturation is not reversible. In other words, if you cool down your egg white after you cooked it, it doesn't go back to being, clear and runny like it was originally. So that's an irreversible denaturation of the proteins. Most proteins don't renature. Once they denature, that's typically permanent. So if you want to kill a cell or an organism, you can boil it or drop it into acid or base, and what you're doing, the reason it kills the cell is that it denatures all the cell's enzymes. And remember I said, and proteins. And remember I said that proteins are critically important to the function of the cell. So if you denature all the proteins in a cell or in an organism, then you kill it, because the proteins do all the work. The proteins are critical. That's what makes life possible. If you denature the proteins, if I boil you, it will kill you. It will work. I'm not suggesting that I would, but boiling kills things because it denatures the proteins in the cells of that thing, and they won't renature. They can't. It's an irreversible process. There are some times, like I gave an example of straightening your hair. So there are some times when you can use heat to unfold proteins, and it lasts a little while, but then they refold. That's kind of what happens when your hair is curly. You can straighten it with heat, and it works for a while, but then you know what happens. It folds back. So most denaturation, like I said, high pH, low pH, or heat is the main thing that denatures proteins. And it's, when you want to purify something, when you want to sterilize water, you boil it. Because if there's bacteria or things in the water, you're not removing the bacteria, you're just killing it, so that when you drink the water, you're just drinking the dead bacteria. And dead bacteria won't hurt you. It sounds gross, but it's safe. So that's, boiling denatures proteins. Proteins are the critical component of a live cell that, the cell can't do without those. So, all right, so let's identify some molecules and then we will be done, I think. Oh, I think I have a little activity beyond this too. Okay, so on this page, let's see if we can identify the amino acid, one amino acid on this page. One amino acid. And remember, you're looking for an amino group, an alpha carbon that has a hydrogen and an R group, and then a carboxyl group, all in a little backbone structure. So the single amino acid is F. F is the single amino acid. So you see the amino group, the alpha carbon in the middle, and the carboxyl group. There is also on this page a short protein, meaning more than one amino acid attached together. See if you can find that one. So that would be I. I has, if you look at it, it's 3 amino acids, NCC, NCC, NCC. That would be the backbone for 3 amino acids. So I guess you could call it a tripeptide or polypeptide. It's a very short peptide, that's for sure. That would be more than one amino acid joined together. All right. So we went over lipids. So we did fatty acids. G was a fatty acid. J was a fat. C was a phospholipid. And then we also went over amino acids and proteins. So F is an amino acid, and I is a short protein, I guess you could say. So my main question is, do you know what A is? Because that's a throwback to the last lecture segment. Can you still identify A? Can you identify B? D? Some of these are the same. E and H. Because I threw in some things from last lecture here. All right, and on this one, let's see if we can find an amino acid on this slide. Let me see if I can zoom out a little bit. On this slide, where's the amino acid? right, I see on number 2. Do you see the amino acid on number 2? There's also an amino acid on number 8. All right, and let's see if there's anything else. I don't think so. I think there's just 2 amino acids. I don't have a polypeptide chain on this one. All right, so let's see. Oh, wait a minute, number 7. Number 7 is an amino acid. That's actually, it has the ionized form of it has the NH3 and COO. Those are both ionized forms, but that's still the amino group and the carboxyl group. So number 7 is also an amino acid. But do you still know what number 6 is? We talked about fatty acids, number 9. Number 10 is glycerol. Do you know what number 11 is? It's supposed to be a phosphate group. They're missing an oxygen there, but do you know what 13 is? 13 is a nitrogenous base. 12 is a simple sugar. 14 is kind of, it's hexagons. So remember, hexagons is a giveaway for glucose. So it's a bunch of hexagons in a chain, so that's a polysaccharide. It's kind of a short snippet of a polysaccharide. 15 is a disaccharide. And 18 is cholesterol. I'm not going to make you identify cholesterol, though. All right, I had something like this in the PowerPoint. I think it's helpful to make a chart like this. Not only do you have to identify the structures, and we have already gone over the different kinds of polysaccharides, but let's talk about just the monomers and the covalent bonds for each of these groups. I think it's helpful, again, to organize it in a chart. So for the carbohydrate groups, the monomers are called simple sugars, or specifically glucose is the main one. And the covalent bond type between monomers for the carbohydrates is called the glycosidic linkage or the glycosidic bond. For fats, the components are glycerol and fatty acids. They don't form polymers, but components. And for fats, the linkage between the components is the ester, E-S-T-E-R linkage, or ester bond. That's the bond that forms between the hydroxyl of the glycerol and the carboxyl portion of the fatty acid. For proteins, the monomers are called amino acids. So make sure you know the structure of amino acids, that you can recognize it. And the covalent bond between amino acids is called the peptide bond. And for nucleic acid, the monomer is the nucleotide, and the nucleotide has components. Remember the pentose sugar, the nitrogenous base, and the phosphate group. But the covalent bond between nucleotides is called the phosphodiester bond. Phospho, di, ester, E-S-T-E-R. All right. So that helps you fill in a little chart like that I put at the end of the PowerPoint. So I would suggest, you make flashcards, you practice identifying the molecules, and make sure you know the characteristics of these different macromolecule groups. It is challenging, I'm not going to lie. So, put the work in, and you may want to find some study partners that you can quiz each other, so that you get to the test, you'll be prepared. And that's the end of this lecture segment.